So now we've seen how you can figure out a DNA sequence and how you can figure out where and when that gene is getting expressed, or multiple genes are getting expressed, but that still doesn't quite tell you what a given gene does. So that's what this video is about. How can we figure out what a gene actually does? And for determining a gene's function, there are two major methods that now get used to accomplish this goal. The first method is a method that's referred to as in vitro mutagenesis. It's also known as creating a knockout mutation. So the way this works is you first figure out what the gene sequence is that you're interested in, and then you create a mutated version of that gene sequence. And then you use one of a range of tricks to add that mutated version back into the organism that you're interested in studying to knock out the normal copy of that gene. So you're replacing a functional copy with a non-functional copy directly. Then you let that organism develop and you examine the result. What's the new phenotype? So this method is a relatively long-term method for studying the effects of a gene. And it's been very widely used, particularly in the model organisms that I've been talking about. The tools that we've been able to use to do this have developed very rapidly just in the last couple of years, which has also meant that we've gone from only being able to do these knockouts in a very small number of organisms, so mostly flies and mice, to now being able to do them in almost any organism. And the thing that's allowing us to do that is what won the Nobel Prize this year, which is this protein RNA complex referred to as CRISPR-Cas9. So this is a tool, we call it a gene editing tool, that contains it's a mixture of amino acids and RNA, and it contains customizable guide RNA that identifies a specific DNA target site to chop. So it lets you target a particular location and disable or change a particular gene at that location. So this method, it's similar to other methods used to create a gene knockout. Basically make something non-functional and then see what happens, and that will allow you to infer what the function is of that particular gene that you're studying. This also poses some ethical dilemmas. So not too long ago, just last year, there are some stories about a scientist who used this method to edit genes in human babies, and this horrified people in large part because there are a lot of ethical concerns associated with doing this type of genetic modification to a person. So again, this is something where humanity collectively has said we need to proceed very slowly with these techniques and carefully with them, and think not just about what's biologically possible, but also what's ethically appropriate with these methods. So that's the first major method for figuring out what a gene does. And again, I want to emphasize that creating a knockout is a long-term process. So typically that's done at the egg stage. You let the offspring develop, and so that offspring is missing that gene, for its entire lifetime. And another technique that is complementary for studying gene function is a technique that's also a relative newcomer called RNA interference. And this method relies on a trick that cells use to identify and destroy foreign RNA. So what biologists figured out is if you can add some synthetic double-stranded RNA that matches the sequence of a particular gene, what that will do in cells is it will trigger breakdown of that gene's messenger RNA. So it's going to transcription might still happen, but if that mRNA gets broken down right away, that mRNA is never going to get processed or translated, so you're not going to get the protein product for that gene. So RNA interference is a really great complementary method that makes it possible to knock down expression of a gene and then see what the consequences are. So, on the short term, on a short time scale, you could do this in a particular type of cell. If you figured out this gene gets expressed in an embryo at this stage of development, then you could use RNA interference to knock down expression just at that stage of development, and then observe the consequences of having done that. So that also, lets you get to the bottom of the matter of, okay, what does this specific gene do? So, in a nutshell, those are the major methods used for characterizing gene function. So the last biotechnology topics that we will touch on are cloning of whole organisms and then using stem cells and examining what types of questions can be addressed using those methods.